So let us start. So before start, again I am welcoming to this 10th Star Open Science Conference 2022. And this is the continuation session of yesterday, 26, session 26. The session is ecological implications and adaptations of microorganisms in the polar environments. So before starting the session, I would like to inform you that each presentation will be 15 minutes for the speaker. Out of 15 minutes, 30 minutes will be presentation and 2 minutes will be discussion. And regarding after this each presentation, we'll have a facility for chat box function for questions. So if anybody asks questions, they can go for chat box function in Zoom. And then session will be recorded to be made available to registered participants in the past events section of the events platform. The organizers are also planning to be made recording to the STAR YouTube channel and website. If anyone doesn't want their presentation to be made available on YouTube or website, they should inform to our IT people STAR 2022 @l1meetings.com before 12th August. And regarding the e-poster, we have a number of e-posters that is also going with parallel session, and that will be available in exhibition e-poster exhibition space on the event platform. So all participants can browse their posters and their leisure and comment on them this exhibition space. So now I would like to ask the first presenter, Jayati Arora, and the title is Exploration of Extremophilic Microorganisms Isolated from the Indian Sector of Southern Ocean for Synthetic Surfactant Degradation Potential. So before this, I would like to request Dr. Sabu, panelist. Please let us inform the timing of the speaker. If it is after completion of speaker, please notice us.
Yes.
Yeah.
Thank you.
Please join the Aurora.
A very good morning to Anil who are present here. My name is Jayati Arora. I'm a PhD research scholar at Amity Institute of Environmental Sciences, Amity University, Noida, India. The topic of my presentation today is exploration of extremophilic microorganisms isolated from the Indian sector of the Southern Ocean for synthetic surfactant degradation potential. Introduction: why are we taking up this problem?
Excuse me.
It's not audible, Jayati Arora.
Volume you have to.
Hello, is not audible, Jayati Arora.
Yeah, we can't hear you.
Is audible?
It's not audible.
Not audible, yeah.
Say something.
Yeah.
No.
I'd like to request IT team, please help us that It's not audible.
Hello.
Ms. Jayati Arora, are you hearing us?
So in that case, we can move the second speaker.
Hello.
Yeah.
Is Jayati Arora?
Yes. Can I start now?
Yeah. Please. Yeah.
Good morning, one and all. My name is Jayati Arora. I'm a PhD research scholar at Amity Institute of Environmental Sciences, Amity University, Noida, Uttar Pradesh. The topic for my conference today is exploration of extremophilic microorganisms isolated from the Indian sector of the Southern Ocean for synthetic surfactant degradation potential. So in introduction, I would like to mention that why are we taking up this problem? As we know that surfactants are used extensively in cleaning industry, especially in the laundry industry. They are being discharged as sewage effluents into the surface waters. And anionic surfactants are one of the most used kinds
I
of
think there is
surfactants.
some audio issue. We are not able to hear.
Hello. Can I continue?
I can hear you fine. I don't know what the other guys.
Okay.
I can hear you. I can hear you too.
So, anionic surfactants followed by the non-ionic ones are mostly responsible for polluting the environment. Increased foaming in the water body, especially in case of Yamuna River in India, we can see the foaming of the river because of surfactant pollution in the water bodies. In this diagram, we are showing the effects of surfactants on the aquatic environment. It causes foaming and reduced dissolved oxygen and disruption of water cycle. And also it leaches into the groundwater and causes groundwater contamination and thus impacts the human health. Here is a description of the effects of various surfactants on the flora and fauna. Planktons, aquatic plants, fishes, all have been shown to be affected by the adverse effects of surfactants. Now talking about extremophiles. Extremophiles are heterogeneous class of microorganisms. They dwell in extreme conditions and they adapt to such conditions for their optimal growth. They possess robust enzymatic and biocatalytic systems and they can be used in various industrial and biotechnological applications. On the basis of the presence in the environment, they can be classified as psychrophiles, thermophiles, acidophiles, xerophiles, etc. The Southern Ocean is an ocean of rich microbial diversity. It is world's largest high nutrient, low chlorophyll region and plays a significant role as a sink for atmospheric carbon dioxide. It harbors microbial diversity, having capabilities of existing in extreme conditions and possessing various different types of enzymatic activities which can be utilized in industrial applications. Materials and methods for my study. I would like to mention that the samples were collected from the Southern Ocean during the 10th Southern Ocean expedition. In the present investigation, 80 samples were taken from water, sediment and algal mats. The samples were collected from different locations from the Bharati Station, Fisher Island, McLeod Island, Brookness Peninsula and Stones Peninsula. All the seawater samples were collected from different depths using CTD operations. Also, plankton and other aquatic samples were collected using multiple plankton net and bongo nets. On-board culturing of microorganisms was done using selective media for further investigation. Till date, our department is maintaining 89 bacterial isolates that have been recovered from various water and sediment samples. As a part of my research work, the focus has been given to the surfactant degradation potential of bacterial species isolated from the Indian sector of the Southern Ocean. The target compound here is sodium dodecyl sulfate, which is an anionic surfactant, also known as SDS. So what we have done is a loopful of the preserved water sample have been streaked on nutrient agar plates to obtain the bacterial diversity, and they have been incubated at 37 degrees Celsius. Then pure cultures were obtained for assessing the SDS degradation potential. Further, the microbes were grown in minimal media. The pH was set at 7, and SDS was added to this media, and the final concentration of 100 ppm was maintained. And the growth of the bacteria was estimated using UV visible spectrophotometer and measured as optical density at 600 nanometers. Further, optimization of bacterial growth was done at various concentrations of SDS ranging from 12.5 ppm to 100 ppm. Also, different pH like 5, 7 and 8 were taken into consideration. Also, different conditions, temperature conditions like 4, 20 and 37 degrees were taken for observing the bacterial growth. This is the data that we have collected for various optimized parameters, that is different concentration, pHs and temperatures. This is the graph that we have obtained from the data that was shown in the earlier slide, and we can see that maximum growth of the bacteria was shown at 100 ppm of SDS, and 7 pH and 37 degrees Celsius was the optimum environmental condition. Now, analysis of surfactant concentration was done using a standard method given by the AFA manual, which is known as the methylene blue active substances assay. This method is based on the formation of a blue-colored methylene blue compound and anionic surfactant, and it is extracted over chloroform, the absorbance of which is measured at 652 nanometers. For the biodegradation of SDS, the setup was made in minimal media, and a pH of 7 was maintained. The SDS concentration was taken at 100 ppm. The flask were incubated at 37 degrees Celsius and stirred at 150 rpm, and the entire setup was for 216 hours, that is 9 days. And the samples were also withdrawn at regular intervals to analyze the concentration of surfactant present in the media. So this is the data that we have obtained. On spiking, the 0 day, we spiked around 100 ppm, and finally after 216 hours, we could see that the temperature, that the concentration came out to be 10.41 ppm. So we can say that around 89.58% of the SDS in the sample was degraded. So we have isolated a species that is able to degrade our SDS. Now we are moving forward for the characterization of the species. For this, we have taken 5% blood agar for morphological characteristics. It was globular shaped convex colonies which were creamy white in color, and they were also gram-positive species. On carrying out the biochemical characterization, we could see that the species is able to grow on selective media weight Parker agar. It was a urease positive and coagulase negative species. These are the results. Black characteristic colonies on weight Parker agar, gram-positive coccus structures, creamy white colonies on blood agar, and positive urease test. We also characterized the species using genetic sequencing. We isolated the DNA, and we performed a 16S gene amplification using the primers, and we got the forward and reverse DNA sequences. On phylogenetic analysis was done using NCBI's BLAST software, and our species SOB1 was found to be 100% similar to Staphylococcus saprophyticus. Major findings of this species is a total number of 48 samples, 48 bacterial samples were assessed for their surfactant degradation potential. The SOB1 species, that is our target species, have been isolated from the same region.
Jayanti, sorry for disturbing. You have 1 minute left.
Yes, sir, I'm about to finish. This species is able to show 89.5% of degradation of SDS, and 100 ppm of SDS was found to be around 10.5 after degradation. This species is a gram-positive coccus, and it is a coagulase negative and urease positive species, and it is 100% similar to Staphylococcus saprophyticus. On concluding, we could say that the study was designed to address surfactant pollution. It possesses 100% similarity with Staphylococcus, and in the future, we are planning the same work with nanoparticles like TiO2 and nanobiochar for achieving better degradation results. Thank you so much. I would like to mention special regards to my Amity University guides, Dr. Anuj Ranjan, Professor Tananjan ma'am, Dr. Abhishek Chauhan, and my guides from NEERI, CSIR, Dr. Reena Mandal. Thank you so much for listening. Any questions?
Mishra, we have 1 minute left. We can take one or two questions.
Sir, shall I ask one question?
Yes, sir.
Yeah, actually, I'm not a microbiologist, but my doubt is that you are telling about its growth, the maximum growth at 37 degrees centigrade. That is
Yes.
right.
Yes, sir.
But this is from Southern Ocean samples.
Right, sir.
But Southern Ocean, where you are getting this temperature?
Sorry?
This temperature, where you are getting in Southern Ocean conditions?
Sir, we have isolated from the Southern Ocean and we have optimized the temperatures at 4, 20 and 37 degrees Celsius for our biodegradation studies. And we are doing an experimental setup and the growth we have seen at 37. We are trying to use multiple parameters like nanoparticles also so that we can see growth in lower temperatures also. It is an ongoing study and we are planning to do further research on this.
So in Southern Ocean conditions, its growth cannot be expected as much what we are getting in 37 degrees. That's right, no?
Yes, sir. So the point is, we are trying to isolate an organism from the Southern Ocean because of their enzymatic activities. We could utilize them in the normal conditions, not in the Southern Ocean conditions, so that we can utilize them for biotechnological applications.
Okay, thank you.
Thank you. Shall I stop sharing?
Yeah. Dr. Mishra, you are muted.
Yeah, thank you, Aruna. Thank you for your nice presentations. So we will not have enough time. So now I can welcome to second speaker, that's Maria Monterio. So the title is Predicting and Validating the Impact of Warming on Terrestrial Microbial System in Antarctic Polar Deserts using Laboratory and Field Space for Time approach. Can start, Maria?
I think I have provided my record. Oh, yeah.
Okay.
Hello, my name is Maria Monteiro. I'm a PhD student from the University of Waikato, and today I'll be talking about predicting and validating the impact of warming on terrestrial microbial systems in Antarctic polar deserts using laboratory and field space-for-time approaches. So the dry valleys are a microbial-driven system. So the effects of climate change on microbial communities will likely affect the functioning of the system. So the first question we ask you today is, how can we study microbial response to environmental changes in the dry valleys? So long-term research has been invaluable to report temporal patterns and variability in the system's climate, hydrology, and biology. However, it takes too long to gather the data and to publish the results. And so considering that climate change is occurring at a fast pace, we need to find complementary ways that enable faster predictions of the impacts of climate change in polar desert ecosystems. And so these lead us to talk about space-for-time approaches, which rely on the use of environmental gradients that reflect past or future changes in the system. And so they act as proxies for the temporal processes that we want to study. Now, it is not uncommon to see studies using distance points along environmental gradients as their sampling design. However, in the dry valleys, the distribution of microbial communities is mostly biologically driven. As such, we need to first describe the variability of these key drivers of communities along the gradients representative of the change that we want to study. And so we define wetness gradients from the shores of 6 lakes in the Wright and Taylor Valley. So 2 lakes, Lake Brownworth and Spalding Pond, they are open basin lakes, which means they have an outflow. Therefore, the water level has been relatively stable. And that means that the wetted areas that surround these lakes on which we define our gradients are also stable. The other 4 lakes, Lake Vanda, Lake Frixell, Lake Hall, and Lake Bonny, they are closed basin lakes. And so the water level is more unstable. In fact, in the past 20 years, some of these lakes has been rising. And so the wetted areas that surround these lakes have also been expanding. And so we hope that by comparing these static transects with these dynamic transects, we will be able to access the influence of historical versus more recent impacts of water availability on the structure and the diversity of microbial communities. So by characterizing the variability of abiotic drivers along these gradients, we were able to define geochemical boundaries that may drive community assembly. And so, for example, we identified 3 geochemical zones by looking at moisture, electrical conductivity, elevation, water activity, and pH. So in wetted areas, for example, they were characterized by having high moisture and low conductivity. On these transition areas, they are characterized by having also high moisture, but much higher conductivity. And then on dry areas, they were characterized by having low moisture and low conductivity. And so we took a sample on a wet zone, 2 samples on the transition zone, and 2 samples on the dry zones. And we replicated that approach across all transects, across all lakes. Now, one of the first results we've got is that we were able to detect the presence of some taxa in wet and dry conditions. So, for example, Federico microbiota, they were consistently present under wet conditions, and the same with proteobacteria. On the other hand, OTUs affiliated with actinobacteria were much more abundant when the soils were dry. And so we can discuss that any changes in these bacterial taxa may reflect changes in water availability. And so these microbes can actually act as biological sentinels for environmental change in the dry valleys. And then we compared the microbial structure along the gradients, and we compare it between the static transects and the dynamic transects. And what we observe is that on static transects, the structural patterns reflect the geochemical zones that previously identified. However, when we looked at the structural patterns of microbial communities along dynamic transects, we observe a spatial segregation of these transition zones communities into a wet-like community or into a dry-like community. So that means that contemporary and historical environmental changes in water availability can be resolved by subtle structural and compositional rearrangements of these microbial communities using geochemically defined transects in static or dynamic scenarios. Now, there is a growing body of evidence that shows that increased human presence in the Antarctic continent is impacting the system. Moreover, when COVID hit in 2019, pretty much all field work in Antarctica had to stop. So perhaps we should try to find alternatives that are not so dependent on field work and could also help to mitigate the impacts of the human presence in the continent. So that leads us to our second question, which is, can we emulate the dry valley terrestrial system and microbial response in the lab? And so to that purpose, our team developed these environmental chambers that are capable of emulating conditions observed in the dry valleys, such as permafrost, temperature, relative humidity, wind, and light. And so we tested these chambers by setting up a wetting experiment. So first we made a permafrost layer, and for that we mixed soils that were previously collected from the dry valleys. They were then sterilized with sterilized water, and we place it in this permafrost container. This container has this cooling coil arm, which freezes the soil. But before the soil was completely frozen, we placed these 4 big cores that were opening both sides when we filled them with more sterilized soil. In one of the cores, we placed these humidity sensors to record changes in humidity from the permafrost to the surface soil. And that's because we wanted to try to mimic sublimation processes that are occurring in the soil. On the top of the 3 other cylinders, we placed smaller cylinders, which we filled with dry soils that we collected close to Lakehore. And so we treated those soils. And so in one set, they were constantly wetted for 8 weeks. On another set, the soils were constantly wetted for 4 weeks, and then they were left to dry for another 4 weeks. And on the third set, there was our control. So there was just dry soil that has never been wetted. And then we incubated these soils under 5 degrees Celsius and relative humidity of 50% and under light conditions as well. And so we sampled communities before the treatment, 1 day after the start of the experiment, 1 week, 4 weeks. At 4 weeks' time, we ceased wetting in 1 set. And so we sample again at 5 weeks, which is equivalent of 1 week of drying, and 8 weeks, which is equivalent of 4 weeks of drying. And when we look at the communities, we observe that the beta diversity changed with wetting. So communities that became wet, they became much more dissimilar from the control. However, when the wetting ceased, those communities that start to dry out, they start becoming much more similar to the dry control again. And then we looked at the response of dominant taxa that were initially present in the dry soils. And so what we saw is that, for example, actinobacteria, which are the dominant taxa in dry soils, their relative abundance decreased significantly with wetting. And the same was observed for acidobacteria. They were quite sensitive to wetting. However, when we look at proteobacteria, they were quite happy, and their relative abundance tripled over the 8 weeks of wetting. Now, when the wetting stopped, what we observed is that actinobacteria started to recover, and so their relative abundance started to increase again. And it was the same for acidobacteria. In fact, acidobacteria increased in relative abundance to values that were close to the control, to the dry control. So the temporal changes in community composition that we observe are driven by this asynchronous response from different taxa present in the dry soils. And to some extent, these compositional changes that we observe, they reflect those compositional changes that we observe in situ using our wetness gradients. More, the communities present in these dry soils are composed by taxa which show different adaptations to water stress, which may reflect the presence of a diverse physiological and metabolic traits and stress mitigation strategies in these communities that are present in these dry soils. Now, it's very important because the conservation of drought-resistant taxa during 4 weeks of wetting can underpin the ability of dry valleys communities to recover from short wetting disturbances. And this is a sign of resilience. That means that these communities can be resilient to short-term disturbance, to short-term wetting disturbances. Now, what we did not expect to observe was the absence of cyanobacteria growth. And that means that probably dispersion mechanisms play an important role in the distribution of this functional group across the continent, as been referred previously in other studies as well. And so this is about a change and studying the impact of change in the dry valleys ecosystem. And change is happening fast. Therefore, we need tools that provide a quick assessment of the thresholds, tipping points, and resilience of microbial communities to increase frequency and intensity of environmental changes. And so we've been analyzing this from a compositional standpoint. However, we need to start asking how these compositional changes affect the function of the system, because they are linked. And so to conclude, I'll conclude by saying that with controlled and adequate infrastructure, laboratorial manipulations can create powerful means of hypothesis testing without the permit, financial, temporal, or logistic constraints, nor the human impact associated to field studies. These studies can be further expanded to include other types of disturbances and can include other types of organisms. Then the results will need to be compared and validated by in situ studies, where ecological factors such as dispersions may influence the diversity and the distribution of communities, and therefore influences the functioning of the system as well. So finally, I would like to thank you to my supervisors, my collaborators, my colleagues, to Antarctica New Zealand, to the University of Waikato for all the support. And I would like to thank you to you too for listening to me today. Thank you.
Thank you so much, Maria.
So we have 1 minute. We have 1 minute
Yeah,
left
please.
for Q&A.
Yeah. Professor Craig, do you ask any question?
I think I pretty much know everything about it.
Hello, Professor Craig.
Yeah, no, I think I know quite a bit about it. Maria is my student. I think I think what the importance of what this study is doing is bringing together the idea that we can work off continent as well as we can work on continent. And I think this is the beginnings of, in putting it out to the community, this is something that we should be considering, given the constraints of working on the continent in the past few years, and certainly what's going to be coming up. Maria, I mean, from a functional standpoint, you mentioned function. What do you see as the next steps in tying function into this?
Well, yeah, well, it's just saying what, in terms of what these compositional changes represent in terms of the function of the ecosystem, because as we've seen, and even in previous sessions, the dry soils, they are composed by these taxa that are almost like in a survival mode and are quite autotrophic. And so based on what we, the keynotes, the presentation by Chris Greening that we've seen last night as well. So they are they are maybe on the survival mode, and but they're also very specialized to these dry and oligotrophic conditions, and without water as well. So, but when we add water, I believe that the whole ecosystem is going to change, because it's going to become much more heterotrophic, I would believe. And so, yeah, but we have to look at whether these functions are maintained by different taxa or by the similar taxa, or yes, in terms of looking at, yeah. So when I talk about whether the functions are maintained, I mean by are they functionally redundant or across from a dry to a wet transect or not, and so, and that of course we're going to have really big repercussions to how the ecosystem function. And the good thing about also, I think, the manipulation is that we can revert the system to what it was before, which is also very hard to do in the field. And so, and this is really important to study, to do more resilient studies in order to evaluate whether communities are able to come back to their original state or not. And so, yeah.
Cool, thanks.
It's not audible. Okay. Yes, Maria, thanks. We can, we are now running short of time. So can we move to the third speaker, Rajani? And the title is Identification of Organic Solutes Accumulated by Two Antarctic Prometheus Strains, Natural Evidence. One is Nuclear Magnetic Response Spectroscopy. Rajani.
Yes, I'm here. Hi, everyone.
Yeah, hi.
Okay.
Can you please start?
Yes, let me share my slide.
Okay.
Okay, can you see my screen now?
Yeah,
Yes,
we can
yes,
see.
yes.
Okay, yeah. My name is Anil
Can you put
Osman.
in slideshow?
Okay, there you go. Okay, my name is Raja Usman. I'm a PhD student under the supervision of Dr. Paradina American from University of Science, Malaysia. I would like to present our research where we identify organic solid accumulated by two Antarctic Pomidesmis strain using NMR, proton NMR. Okay, so cyanobacteria or blue-green algae are known as prokaryote photosynthetic microorganisms that are featured among the pioneering living organisms on planet Earth. They have been in existence for over 3 billion years and are the main primary producers of organic matter. Their ability to perform oxygenic photosynthesis makes cyanobacteria among the first organisms to have released oxygen into the then primitive atmosphere. With regard to distribution, cyanobacteria has a cosmopolitan one. They inhabit almost all kinds of habitat, from Antarctic lakes to thermal springs, arid desert, and tropical acidic soil. This versatility exposes cyanobacteria to varying environmental conditions. As a result of that, they can also produce an array of metabolites that are found to have anti-tumor, anti-cancer, antibacterial, and antibiotic activities. Production of this metabolite is sometimes dependent on the stress condition they found themselves. So with regards to salinity, salinity is one of the controlling factors of cyanobacteria, affecting their photosynthesis, function of plasma membrane, therefore affecting the cell growth in general. Salinity also exerts toxic effect to the sensitive cyanobacteria, thereby restricting their distribution to certain environment. For cyanobacteria to survive in changing salinity, they have to evolve a mechanism of salt adaptation or salt acclimation. And they achieve that through two principal strategies. One is ionic regulation, and the other one is osmotic regulation. Ionic regulation involves the uptake of salt into the cells via transport, into the cytoplasm via transport, while osmotic regulation involves the synthesis or accumulation of compatible solids. These compatible solids are small molecules that act as osmolyte, preventing the cell from osmotic shock. This compound can either be synthesized by the organisms themselves, or they can be accumulated from environment when other organisms made them available. So as a result, based on this, cyanobacteria can either be classified into halotolerant or halophiles. So why we are interested in doing this is because cyanobacteria in Antarctic ecosystem are found to be the major component of most mud in most Antarctic habitat. And these habitats experiences varying salinity, fluctuations of salinity, especially during the winter and summer season. The formation of ice led to the formation of hypersaline, concentrated hypersaline layer below the ice. And cyanobacteria are also found to be surviving under this highly saline environment. Freshwater also in Antarctica can become saline as a result of excessive evaporation and less precipitation. Also, most habitat in Antarctica also receive saline as a result of wind blown from sea. So the formation of this varying saline condition could provide an important ideal biological system for studying how cyanobacteria adapt to salinity in Antarctic habitat. Today, little is known about the physiological response of Antarctic cyanobacteria to salt strain. So in our current work, we present the study on the growth profiling, carbohydrate accumulation, and lipid pit stop in 2 formidesmic strains grown under varying salt concentration. We also employ the use of proton NMR for identification of the organic solute involved in their salt acclimation strategy. So the 2 strains used in these studies were originally isolated and characterized and deposited in our culture room. The organisms were previously collected, the match were supplied to us by Dr. Paul Brode, and the samples were collected in the 70s. So one of the strains, CR147, is collected from freshwater environment. CR147 is collected from a little bit saline environment. So the cells, the strain were maintained in BG11, and we use the BG11, the culture as a scale-up culture for our experiment. So we grow them in 4 different conditions. We use only BG11 to defeat freshwater condition, and we also modify BG11 by addition of appropriate gram of sodium chloride to defeat 15 ppt to defeat brackish environment, 35 ppt to defeat sea salinity, and 65 ppt to defeat hypersaline condition. So from the scale of culture, we inoculate a known amount of biomass into this media, and we assess their growth under varying salinity. We use optical density and chlorophyll A as a proxy for the growth analysis. And then at the end of the experiment lasted for 30 days. So at the end of the experiment, we harvest the culture and access the carbohydrate feedstock and protein feedstock. For the determination of compatible solid, osmo-adaptive compound, we prepare a mass culture and we grow the organism in 35 ppt. Both the 2 strains were grown in 35 ppt. And we harvested them at day 30 and combined the culture. And we extracted the compound using 3 methods. One using 80% ethanol, and the other one using sterilized distilled water, and the third method using methanol, chloroform, and water. The extraction product were freeze-dried, and then the freeze-dried extracts were suspended in deuterium water and analyzed using 700 NMR spectrophotometer. So here is our result for the growth profile. And the first pictures here is the cultural morphology in various salinity. And as we can see here, as the salinity increases, the chlorosis also increases. The color is fading away as the salinity increases. This is telling us that the salinity is having effect on the growth and biomass accumulation in this compound. And if we look at this graph, the A is for CR11, B is for CR147. If we can see here, the red is the control media, green is the 15 ppt, while the blue is 35 ppt. The black is the 65 ppt growth condition. So if we can see here, the first 3 conditions, 0, 15, and 35, the difference, the growth is not much significantly different, but growth is significantly affected as the salinity increases to 65 ppt. While CR147B here, at least they have performs better than CR147 because even at 65 ppt, growth is reasonably maintained. And both chlorophyll A, optical density, and chlorophyll A shows almost the same pattern of growth. With regard to the carbohydrate feedstock, the figure A is for CR11. As we can see, the increase in salinity has no much effect on carbohydrate production, but the carbohydrate production spikes a little bit at 35 ppt. In B, in CR11, strain CR11, it is evidently shown that increase in salinity increases the production of carbohydrate, which is part of the sugars that help cyanobacteria to survive in saline situation. But with regard to
Suraj,
the,
sorry for disturbing. You have 1 minute left for.
okay, with regard to protein, it shows a little pattern, increase in CR11 and then decrease in CR147. So here, we managed to identify two compounds. One is trehalose in CR11, and then glucosyl glycerol with some traces of sucrose in CR147. It is not surprising that because CR11 is isolated from freshwater environment, and freshwater cyanobacteria are known to produce sucrose and trehalose in response to salinity, while CR147 is isolated from elite high saline environment, and strain of cyanobacteria with moderate salt tolerance are known to produce glucosyl glycerol, and both extract spectra is the same in both extract. So ongoing experiment here, we are trying to analyze the effect of salinity on the concentration of this osmolalite that we discovered. And then another project is underway genomic project is underway to investigate the genes responsible for salt acclimation. So in conclusion, based on this, we understand that exposure to salinity, cyanobacteria from Antarctica can adapt to varying sodium chloride concentration. The study provided an opportunity for identification of compatible solid responsible for osmotic adaptation produced by Antarctic cyanobacteria for the first time. Our genomic studies, when completed, will add knowledge to address one of the scarce questions: how can changes in form of and frequency of extreme events can be used to improve biological understanding? Another question can also be addressed: what is the genomic basis of adaptation in Antarctica and South Ocean organism and community? This study will provide further insight into salinity tolerance. Okay. I would like to acknowledge my supervisor, Federico American, and other team members. Thank you all for listening.
Thank you, Suraj.
Okay.
It's a nice presentation. So I have one question.
So, shall I ask one question?
You checked the salinity with two different strains,
Yes.
right?
Yes.
And you checked 0, 15, 35, and 65.
Yes.
Why you selected only these 4 salinities? And what about the in-between salinities? Have you checked in between the salinities of their biomass changes or anything else?
Yes. In the preliminary studies, we expose these trends to various salinity to see the maximum salt tolerance level that they can survive. So the two strains have varying salt tolerance. Strain CR11 can tolerate up to 65 ppt, while strain CR147 can survive until 105 ppt. So we selected 0 to represent freshwater condition, and then in between from anything above 0 until 25 to 28 represents brackish condition. So we just select 15, and then we also select 35, then C salinity, and then we selected 65 to present a little bit hypersaline condition.
Okay, then another question.
Yeah, I have one question.
Yeah, please.
Actually, you are telling about the cyanobacteria adaptation in the Antarctica conditions. Freshwater is becoming saline because of evaporation. But I don't think evaporation in Antarctica conditions it's happening like that, because this maybe cyanobacteria is coming from the melting, and the freshwater is coming and it is mixing with the saline water and it is becoming saline. That may be the condition. Can you just clarify that?
Excuse me? No. Excuse me?
Did you
Pardon?
hear me?
No, I didn't hear the last part.
Yeah, actually, I suggest that it is not because of evaporation. It may be because of the cyanobacteria in the freshwater. Due to melting, cyanobacteria is coming and the freshwater is mixing with the saline water. in the Antarctic, Southern Ocean conditions, and the growth, it is getting the growth. That may be correct, not because of evaporation. Am I right?
Okay. That one is part of it, but also, because there is less precipitation in Antarctica, the fresh waters can evaporate. So when water evaporates, it making the remaining water a little bit like saline.
But evaporation is happening in Antarctica. It may be because of mixing with saline water. I think so.
Okay. I will check this.
Okay. We will if we have a time, we'll discuss much at the last. Last of our presentations. So can I invite to Melanie and Deloche? Yeah, Melanie Deloche is there.
Could not find her in this panelist list.
Yes, Dr. Sabu.
I could not find her actually. That name is not there in this list.
So then we can move to next speaker.
Next, yeah.
Sir, we have just promoted Banana Deloche as a panelist. She has joined.
Yes. Okay. She's
Melanie Deloche is there.
Yes, just now.
Yes, I'm here. Sorry, I didn't have the link. So I will share my screen. Can you see them? Can you hear me?
Yeah, it's okay. It's fine.
It's okay? Okay.
Yes.
So good morning, everyone. I'm Melanie Deloche. I'm from the Millennium Institute of Biodiversity of Antarctic and Subantarctic Ecosystem from Chile. And today I will present you how the level of habitat specificity can modulate the microbial biogeographic patterns in the Southern Ocean from community to population scale. So biogeography is the study of the distribution of biodiversity over space and time. In the Southern Ocean, it has traditionally investigated the geographic distribution of microorganisms, and those studies reflect the important role of Antarctic circumpolar current as a connector of biodiversity and the role of fronts as a biogeographical barriers. But what about microorganisms? So until now, microbial biogeography have been mostly studied at the whole community level without considering the microevolutionary process that are behind the observed microbial biogeographic patterns. These microevolutionary processes are genetic drift, dispersion, selection, and diversification, and they act at a much finer taxonomical scale, which is the species or population level. So there is a need to consider the intra-operational taxonomic unit diversity to assess and to decipher which microevolutionary processes are behind. In addition, different factors can influence the biogeographic patterns, such as the spatial scale, the microbial guild, or the habitat heterogeneity. So we choose to focus on the habitat heterogeneity and how it can modulate the biogeographic patterns. So to obtain different levels of habitat selectivity, we choose 3 different habitats from sediment to benthic host associated habitat. And our host model was the Abatus sea urchin, which is a deposit feeder. And different species of this sea urchin exist in the Southern Ocean, but they all have similar ecological features. And 2 habitats were associated with the host, the gut content, which is the sediment filling the gut, and the gut tissue. So our main hypothesis was that the degree of habitat selectivity can be considered as a proxy of the dispersal potential, modulating the microbial biogeographic patterns observed across the Southern Ocean from population of the whole community scale. So we hypothesized that microorganisms from the sediment are more likely to disperse than the ones associated with the gut tissue, and this will result in weaker spatial patterns for microorganisms from the sediment. So our main objective was to assess the impact of the habitat selectivity on microbial biogeographic patterns across the Southern Ocean, and this at different ecological levels, from community to intra-operational taxonomic unit. So to do so is simply took place across 4 regions in the Southern Ocean, in Patagonia, in Antarctic Peninsula, in South Georgia, and in Kerguelen Island. Some of these sites are connected through the Antarctic circumpolar currents, and some of them are separated by the Antarctic polar front. So in each of these sites, habitus specimens were collected, as well as their surrounding sediment. Some environmental benthic data was also acquired from bioarray. Then we applied a conventional metabarcoding approach, and the bioinformatic processing of the sequences took place at 2 main levels, to the community, by clustering in OTUs, and at the intra-OTU level using the oligotyping pipeline. Briefly, these pipelines allows to identify true biological meaningful variants of sequence that we name oligotypes, that by overcoming sequencing errors. So today I will present you the main results at the community and at the intra-OTU level. So first of all, at the community level, in this principal coordinates analysis, we see that different regions harbor different communities assembly, that the region effect was twice stronger than the habitat one, and that the major difference observed were between Antarctica and Patagonia. This suggests that bacteria communities displace biogeographic patterns reflecting the role of the Antarctic polar front as a barrier to bacterial community homogenization. But the effect of the region can be either due to the spatial distance that will limit the dispersal, or through difference in the environmental conditions between the regions. So most distant regions will exchange less microorganisms, while more than the more closest ones, and this will result in more or less similar communities, while different regions that have different environmental conditions will imply different filtering, environmental filter. So to disentangle those effects, we did a partial mantle test, and the results show that the geography effect was stronger than the environmental wall in the community dissimilarity, and this suggests that microbiota is constrained by both environmental filtering and dispersal limitation, but that the effect of the dispersal limitation is stronger. But if we now go back to our gradient of habitat selectivity, in this principal coordinates analysis, we see that communities from the sediment are more homogeneous between within a region and more heterogeneous between regions, and the opposite is observed for the gut tissue communities. And this explains that the region contributions decrease along the gradient of habitat selectivity, and the same pattern is observed in this distance decay relationship, where we see for each of the 3 habitats that the greater the geographic distance and the more different the communities are. So these results suggest that habitus gut tissue homogenize the microbial diversity between biogeographic regions by imposing different filtering conditions. By imposing filtering conditions that constrain the microbial diversity present in the surrounding sediments. So I don't have the time to enter in detail of the OTU scale, but this difference in the community compositions are in part explained by the presence of region-specific OTUs that characterize each biogeographic region. But we also identify common OTUs to all the regions and what we call the geographic core. And we focused on these OTUs to assess the bacterial biogeographic patterns at a much finer taxonomical scale, which is the intra-OTU level, by applying common approaches that are used in microorganisms phylogeography. So for each of the for each habitat, we selected the most abundant OTUs from the geographic core, and we applied the oligotyping pipeline, and we construct networks as this for each OTU. These networks shows the abundance and the difference between oligotypes and their distribution around the sampling sites. So based on this network, we calculated different genetics index that, such as the phylogeographic structure index. This index is based on the genetic difference between oligotypes. It varies between 0 and 1, 1 meaning high phylogeographic structure, which means that different regions are characterized by different oligotype sequences. So we test this index in between the different habitats, and we highlight that the OTUs associated with the gut tissue habitat had higher phylogeographic structure than the one associated with the sediment ones. This suggests that OTUs from the gut tissue are more constrained by biogeographical barriers than the ones associated with the sediment habitat. In other words, that OTUs from the sediment habitat are more likely to disperse through the Antarctic circumpolar currents than the one associated with the gut tissue, permitting an homogenization of the microdiversity. In summary, our experimental design enabled to compare the microbial biogeographic patterns along a gradient of habitat selectivity, and this at different scale. So we showed that at the community level, the geographic differentiation was stronger for the sediment than for the gut tissue habitat, highlighting the important role of the Antarctic polar front as a biogeographical barrier and the homogenizer effect of the gut tissue habitat. But once we consider common OTUs to all the regions, we show that the phylogeographic structure was stronger for the gut tissue habitat than for the sediment habitat, suggesting a higher dispersal capacity through the Antarctic circumpolar current for OTUs associated with the sediment. Finally, our study highlights the importance to take into considerations the habitat specificity as well as the ecological organizational level to assess the microbial biogeographic patterns. Thank you for your attention. Thank you to Millennium Institute BASE, and I will be glad to answer all of your questions.
Thank you, Melanie Deluge. Is a nice presentation on microbial biogeographic patterns in different region. You got a different community and their activity and all these things. So may I request to this panelist or this audience who can ask any questions regarding.
Can I ask one question?
Yeah, we have in chat box.
Okay, check chat box.
No, we don't have any question in chat box. Yeah, please.
Yeah, so you are telling that the Antarctic polar front is acting as a barrier for this.
Yes.
So polar front is little away from the Antarctic coastal regions and all, and two more fronts are coming there. That is South Antarctic circumpolar current front and the southern boundary. Whether these two fronts are making any barrier for this or not? Did you study on that aspect?
Sorry, can you repeat? I didn't understand.
Yeah, two more fronts are there when we are moving towards the Antarctica. That is southern Antarctic circumpolar current front and southern boundary. Whether these fronts are acting as barriers for this or not, whether you have studied on this.
So the Antarctic polar front acts as a barrier because there is a difference in the thermocline, and we show that these differences are observed as well as at the community than at the intra-OTU level. There are also we also evidence them at the OTU level, and I don't know, I didn't.
Okay. Thank you. Okay.
Okay,
Yeah, thank you.
Thanks, Melanie. Any questions?
I have one. Could I just come in and say thank you?
Yeah, David. Yeah, David, please.
That was a very nice talk, very interesting study. And what strikes me about what you've presented is how similar the observations you're making about microbial biogeography are to other substrates and species that are starting to emerge around the continent. So it's nice to see similar spatial patterns starting to emerge in microbial biogeography in the Antarctic.
Yeah, it's very interesting that the same patterns are observed as well as the microorganisms level that are the microorganisms, and they are all governed by the same microevolutionary process.
Okay, thank you all. So if no more question, then I can go for the next speaker. That is sixth speaker, but sixth speaker is absent due to some issue. So then we can go to seventh speaker. The seventh speaker is Luke Copton, and the title is Bacteria and Ice Nucleation Bacteria in the Antarctic Atmosphere. Hello.
Hello.
Yeah, please.
Hello.
Please, Luke.
Yeah. Okay, so hello everybody. I'm Luke Cockton. I'm a PhD student at Northumbria University. I'm working with Professor David Pearce, and today we're talking about bacterial ice nucleation in the Antarctic atmosphere. Okay, so just firstly then I'll give you a little bit of background. So bacteria in the atmosphere tend to range in abundance from 10 to the 2 to 10 to the 6 cells per cubic meter. And this is affected by factors such as wind direction, wind speed, and air temperature. In terms of diversity, there's been more than 150 different genera of airborne bacteria that have actually been sampled and analysed from both terrestrial and marine environments. And in the Antarctic, so taking air samples along the coast has actually revealed the presence of different microbial communities at different locations, which is quite interesting. And as well as being present in the atmosphere, bacteria have also been shown to be metabolically active, and they can influence climatic processes such as cloud formation via ice nucleation activity, which is what we'll be talking about today. Okay, so what are ice nucleation bacteria then? So right now there's been more than 20 bacterial strains that have been identified as ice nucleation active, and the majority of these are gram-negative bacteria. They all contain a gene which encodes the ice nucleation protein, which is around 1,200 to 1,500 residues. And this anchors to the outer membrane. It also forms aggregate structures. So the most active ice nucleation bacteria, which basically means the ice nucleation bacteria active at the highest temperature, so up to -2, have the largest ice nucleation sites. An example of this is Pseudomonas syringae, which has the INA Z protein. And within the actual protein structure itself, a key region is the central domain, which has 16 residue repeating motif and forms hydrogen bonds with water molecules via serine and threonine. So they've actually been isolated and studied from polar regions, specifically Pseudomonas antarctica, which is isolated from Ross Island. Okay, so why are we looking at this then? So we're looking at this because aerosol particles have a big role in influencing the temperature required for freezing. So, and the number of ice crystals that are actually present within a cloud influences the cloud's radiative properties. So this basically means the ratio of reflection and absorbance of incoming and outgoing radiation. And ice nucleation bacteria, importantly, can catalyze this process at temperatures as high as -2. So in the atmosphere, a big particle source of what's known as heterogeneous ice nucleation is mineral dusts and clays, but they don't catalyze this process as high as ice nucleation bacteria. So there's a potential for studying this and integrating this into climate models. And today there's only been a handful of climate models that have actually added data on this, and they've always used sort of simulated scenarios where you're inputting vast different, but vastly different numbers basically, and concentrations that are actually present in the atmosphere. So the goal of this study was to expand on this and gain some data regarding iron concentrations. And this is especially important in the polar regions. So the specific aims were to quantify the copy numbers of 16S rRNA, which is universally expressed in all bacteria. So that'll be a measurement of total bacterial cells in the atmosphere, and to actually study the presence of the isonucleation gene itself per cubic meter of Antarctic air at a range of marine sampling sites. We wanted to determine what percentage of the total bacteria actually have an IN gene. So we'll look at the total number of 16S and the total number of an IN and get insight into that. Then we want to look at how variable this actual percentage of the total population is over time. So in terms of sample collection, so they were collected on board collected during the ACE cruise, which was a 90-day expedition, which basically went all the way around Antarctica from December 2016 to March 2017. So we had an air filtration set up on board the ship, which had a flow rate of 20 litres per minute. So it basically pumped air and then it captured these cells on the top of a filter membrane. So each sample was ran for 12 hours. So meaning that each sample consisted of over 14 cubic meters of Antarctic air filtered on different sampling days. Some were taken in the morning, some were taken on the evening. These were then sterilely kept and then transported to the lab, where we DNA extracted from these filter papers using a commercially available kit. So because we were dealing with filter papers, some steps to improve yield were included, such as cutting the circular filter papers in thin ribbons. So this is important because when you actually have the filter papers in the tube, you need to avoid clusterings to actually get the cells off the filter paper. So one way of doing this is just to increase the surface area. We also increased the volume of lysis buffer and added a water bath sonication step after the bead beating step at 65 degrees, 30 minutes, which has been previously shown to increase yield. And all these steps were performed in a class 2 hood, reduce contamination, which is a big issue when dealing with low biomass air samples. So this is just a map showing the trajectory of the cruise ship. So the samples that were analysed in this study were taken along the first and second leg of this cruise. So from the 22nd of December to the 28th of February. And then this is the ship that the samples were taken on. So the W1, W2, W4 just show the different locations on board the ship where we obtained the samples. So all the samples in this study were taken on at the W1 location, which is towards the front and higher up on the ship to reduce contamination. We also had backup samples at W2, and interestingly, we had samples at W4, which is at deck height. So the idea there was to capture any marine spray bacteria, so they'll be analysed later. Okay, then, so how do we study the genes of isonucleation bacteria and total bacteria? So we use the quantitative polymerase chain reaction. So this is just essentially like a normal PCR where the target molecule increases exponentially each cycle, and we can actually monitor that real time via an increase in fluorescent signal. So we'll essentially have DNA standards. So we'll have run, say, 1 million copies of the IN gene in the first standard, the serial diluted down, and then the higher the starting quantity of the gene, the lower the signal threshold. So we can actually plot a nice standard curve, and then we'll extrapolate from that our unknowns, so all of our S samples. So we used 16S primers, 27F mod, and V519 R mod bio. These span the V1 to V3 region of the 16S gene. And then for iron quantification, we use pre-published iron primer pair. These were basically designed on the manual alignment of 20 sequenced iron genes. They targeted a conserved region within block 4 of the gene, which has reduced imperfect pattern of homology. So the idea with this is that it was conserved and it allowed the production of a single PCR product. And then importantly, to control steps were melt curve, agarose gel electrophoresis, and Sanger sequencing of products. So we ran our PCRs and cut out the bands that were at the right molecular weight and Sanger sequenced these to confirm these were in fact IN genes instead of artifacts or background amplification, which is a big concern because we're working with things that are very low biomass and also studying functional genes. So it's really important we know that the signal is indeed above background. So this is a big challenge throughout working with this. We had to test multiple different universal primer pairs for the IN gene, and yeah, so we also optimized the 16S method using 0.2 as the primer concentration. Okay, so this is just a graph showing, the point of this graph is to show that we have an apparent increase in 16S copies numbers throughout February. So what this graph is showing on the y-axis is the copy numbers of the 16S gene per cubic meter of Antarctic air, and then on the sample date that was taken. We have the same picture for IN. So that what's interesting is they tend to follow a similar trend. So the highest IN at the top right of the screen there was also that's the second highest 16S copy numbers. And the IN also follows this increase throughout February trend. So we also plotted a graph of variation of IN percentage. So the majority of samples, the IN percentage of the total population tended to range within this 0.001% to 0.5%, so this bottom line on the graph. What's interesting is when we go above this percentage, it tends to be on consecutive days. So the first occasion was the 26th of January, so we see a 3% of the total population, followed by an additional 3% on the 27th. This is also present on the 9th of February and the 10th of February. So this suggests a higher IN percentage of the total population on consecutive sampling days. So just to summarize then, so the data that we collected was the mean 16S, so total bacterial copies per cubic meter was around 800,000, and for IN it was just over 100. And they both show a similar temporal trend with an increase in February. And the IN percentage of this total population tends to fall within the 0.001% to 0.5%. And where we go above the 0.5%, it appears as clusters on subsequent sampling days. We're also working with Arctic air samples using the exact same methodology to establish an Arctic data set. So we have some preliminary data with snow, soil and air samples. And I'd just like to thank everybody for listening and the conference organisers for giving me the opportunity to present my work. I'd like to take any questions. Thank you.
Thank you, Luke, Doctor. It's a nice presentation on this bacteria and this ice nucleation in Antarctic atmosphere. So, may I ask people to have any question?
Hi, Luke. Thanks a lot for a very nice presentation.
Yeah, hi, Tina. Yeah, please.
Very interesting. I was wondering, what do you think are the sources of these INA genes? Because the bacteria that you target are actually terrestrial in origin. They are not usually found in marine sources, and it's a cruise that you did. So do you think they are coming from the Antarctic continent? Or?
Yeah, I don't know. So it's interesting because when we see these higher spikes of iron percentage, it's not as if we're near a port. It tends to be sort of while we're out at sea. So we really need to get more data on that and then coordinate that with the meteorology data to find out the prevailing wind direction then. But it's most likely from the main continent. But yeah, we need more data on that, especially. It will also be good to get the data from the Arctic to see what the story is there and where the higher percentages are there as well. But, yeah.
Hey, Luke, a quick back trajectory analysis would give you a really good indication whether or not the air was transiting recently over the continent. That would be a really good jumping off point for the IN. Yeah, the other thing, I wouldn't mind a bit of clarification on the cleaning protocols and the controls used. So you've got a general increasing trend of IN16S. How often and how did you clean your sampling rig? I presume it's the same one you're switching out filters. Was that cleaned in between, and how? Because IN apparently, according to Paul de Motte's group, Windex is the best thing to get rid of IN, of course, for 16S for DNA contamination. You want to use something like hydrogen peroxide or chlorine. So trying to do both IN and 16S at the same time can be quite challenging.
So I didn't take the samples myself, so I've analysed these samples. But we have controls, and they are not for IN and for 16S. They're not above the background of the assay, so they blended in with the kit negatives and the non-template controls of the PCRs. So that, yeah, that's a short answer, really.
Okay, so I think there is no more questions. So I think one is from chat box, that is Pedro Libri. So that is, the question is, did you analyze the general microbial composition of the aerosols you collected as well?
Sorry, could you repeat that? Sorry, I couldn't hear the question there.
I can reread it, Luke. So did your analysis also study the general microbial composition of the aerosols you collected?
No. But it's definitely interesting. There's lots of sort of different things that we could look at in the future. It's just important to mention this is still sort of an ongoing study, so it's not finished. But yeah.
Okay, thank you. So then we'll move to the last speaker. Of course, she is a panelist, I think, Tina, Santl-Temkiv. So the title is Marine Emissions Dominate Bioaerosol Population in Coastal Antarctica. So
yeah, thank you very much. Let me just share my screen. I'm going to, and thank you very much to let me present in this session. I'm going to present some preliminary results from a campaign that we did in 2019. It was the PICE project led by Manuel Dalosta and at the Spanish research station in the on the Livingstone Island. So the campaign actually had the cruise part and the station part, and I'm presenting now the results from the research station. I can link to some of the other talks in terms of motivation. So Luke already introduced the topic very well. Why we are interested in biological aerosols. So aerosols have a lot of climate impacts that are both direct impacts through interaction with radiation, and indirect impacts through the impacts that they have on cloud development, precipitation development. So clouds reflect solar radiation, have a cooling effect. They also have a warming effect where they entrap infrared radiation. And biological aerosols are known to impact cloud development, precipitation development, and therefore the lifetime of clouds. From all the different types of aerosols, we have aerosols that form in the atmosphere, the so-called secondary aerosols, which form from volatile compounds by condensation. and primary particles which get emitted as particles into the atmosphere. And biological aerosols are part of these primary particles, and they can present quite a large fraction of total aerosols, both in number and especially in mass, because they can be important in this large particle size range. And these are a diverse set of particles composed of microorganisms as bacteria, fungi, dispersal units as pollen, different types of spores, but also compounds that can either be in the atmosphere on their own or attached to inorganic particles. So it's very diverse, and that makes it difficult to assess. Antarctica is a special case because when we look at the Arctic, the air masses are entrapped in this polar dome, which during the summer contracts and is dominated by local sources, while during the winter and spring it expands. And therefore there is a large contribution from distant sources. But in Antarctica, the air masses are quite isolated because of this circumpolar vortex, which is a system of low pressures that represents a barrier to long distance dispersal. So local sources are probably more important for Antarctic bioaerosols compared to the Arctic. So we studied bioaerosols, and as I said, it was on Livingstone Island, as you can see here, and we used two approaches. One approach was a real-time instrument that has quite a high flow rate compared to some other instruments of this type. So it sucks about 30 liters per minute, and then quantifies coarse particles between 1 and 10 micrometers, as well as fluorescent particles based on fluorescence of NADH and riboflavin. So this is it gives you a very well time resolved information on bioaerosols, but not much information on what type of bioaerosols these are. But that's why we combine this with collecting samples for offline analysis. And this was from it was all collected in proximity of the research station. So nearly the same location. We used this high flow impingers that was situated high up on a large outcrop. And these have a very high flow of 3,000 liters per minute. So you can really collect a large volume of air, or aerosols from a large volume of air in a rather short time. So we collected 14 bioaerosol samples over these 3 weeks, each from about 1,000 cubic meters. Then we also collected some fresh snow samples and potential source samples, so marine samples as sea water, sea surface microlayer, some sea ice, but that was from further away, and soil samples. We then co-extracted DNA and RNA. Aerosols are collected in a preservation solution, so we preserve RNA and can also do more activity-based studies. But right now I will show the results based only on DNA analysis. And then we use qPCR and amplicon sequencing targeting 16S rRNA genes and molecules, and then the data to pipeline to analyze the samples. So here are the results on the quantities of bioaerosols over time. So in dark blue, it's the cumulative total coarse aerosols, and it's a log scale, so about 10 to the 6 aerosols per cubic meter of air in this coarse range. And in this cyan color, you can see the bioaerosols, and you can see that the concentrations are extremely low, and they represent a very low, small fraction of the total aerosols. In orange, I plotted the qPCR data, which agrees to some extent with the real-time measurements. So there were around 10 to the 2 bacterial aerosols per cubic meter of air, very, very low concentrations. When I compare that to our measurements in the Arctic, the concentrations in Antarctica are much lower, both for air samples and for snow samples, as you can see here. This orange line represents the detection limit, and often with the molecular analysis, we were below the detection limit. So the concentrations were very low, and these samples are very tricky to work with. I also plotted rain events here with this blue line, and you can see that the concentration of bacteria falls below the detection limit after this large precipitation event, which is also something that we have observed in the Arctic. So kind of a washout of bacterial cells with snow. We looked into the impact of meteorological conditions on bioaerosol concentrations, and we saw some correlations. So the temperature correlated negatively with the concentrations both of total and of fluorescent aerosols. And also there was a weak correlation between precipitation and the concentration of fluorescent, so bioaerosols. The correlation, a negative correlation with temperature is rather unusual. It has been observed previously in temperate regions, but in totally different temperature range. But there is a study that shows that from marine environments through this bubble bursting process, a lower temperature, which are the blue dots, will give a much higher concentration of aerosols. So almost 2 orders of magnitude more when you decrease the temperature to 0 degrees compared to 19 degrees. But this is of course a temperature of the water column, and we measure the air temperature. So the question is whether that could be the explanation, that these marine aerosols are aerosolized better at the low temperatures. Regarding the precipitation, we recently published some data from the Arctic where we show that the precipitation event, here marked in blue, will wash out bacterial cells from the atmosphere. So, sorry about that. So while we detect bacterial cells prior to a large snow event, we detect nothing for several weeks after the snow event in the Arctic. And perhaps this is a similar observation that we have here in Antarctica with this impact of precipitation. Looking at the community composition, here is an NMDS plot with all the source and atmospheric samples. And you can see the soil samples here and then the marine samples here. And the air samples are a bit scattered, but they are, many of them get quite close to the marine samples. So we see that the marine samples are pretty influential for these bioaerosol populations. And that has been confirmed with a source tracker analysis, where we see that around 20% of ASVs in these air samples are associated to marine sources, while a much smaller fraction comes from soil. And then there are a lot of unknown sources, indicating perhaps, of course, we have not captured the full diversity, but probably also there is some longer distance transport. What are these taxa that are associated both with marine and with air communities? They belong to Flavobacteriales, and in terms of genus, it's mostly these Polaribacter, which are associated, they are heterotrophs associated with algal blooms. So they are very dominant in these post-bloom situations. And one can see that they are really emitted. So these are the air samples and these are the sea samples, and you can see that they are pretty predominant. While, for example, if you take another typical marine taxonomic group like the SAR 11, they are predominating the marine samples, but they are not that predominant in air samples. So there is a preferential aerosolization of these Polaribacter types. Another study that came out of this campaign did some air mass backward trajectories and clustered them. So they describe 5 clusters of these air mass trajectories. And we try here to look into how the particle concentration vary for these different air mass contributions. And you can see the results for the fluorescent particles here and for the total particles here. We see that some of the clusters coming more from the southern regions have a higher concentration of fluorescent and total aerosols, while these cluster 5 and 4, they are lower in concentrations. And in this Breen et al. Study, they also look into using satellite images, what type of environments these trajectories pass, and group the environments into open sea, marginal ice zones, and sea ice. And so we can see that when the trajectories pass less of these open sea areas, but more of these marginal zones and sea ice areas, we get a higher concentration of total coarse and bioaerosol particles. So just to summarize, in the end, we see very low concentrations of both fluorescent aerosol particles and bacterial cells, about 100 per cubic meter, so very, very low compared to other environments. We see that temperature and precipitation negatively correlate with fluorescent aerosol particles. We see, based on bacterial community composition, that they are that the airborne communities are closely similar to surface microlayer and the seawater. That is confirmed by source tracking. And we see that there are specific taxa associated with this relationship, and in particular, this bloom-associated Polaribacter genus. And then we also see that there are high concentrations of total and fluorescent coarse aerosols associated with air masses passing areas with higher fraction of sea ice and marginal ice zones. So thank you very much for your attention. And I also want to thank, of course, the logistics support at the Juan Carlos research station and the whole PI's group and the funding that we received. Thank you.
Yeah, thank you, Tina. Thank you. It is really very informative of this bioaerosol population in coastal Antarctica. So I would like to ask our people and our panelists or the speaker audience, anybody have any questions?
Yeah, I have a question. The Polybacter, why does it preferentially aerosol? What are the properties of Polybacter that why does it preferentially aerosol?
So in this process of bubble bursting, often it's considered that it's the surface properties that are important, because you have small air bubbles entrapped in the water column. And that is an air-water interface where more polar compounds or particles will partition in this interface and then, get carried up to the sea surface and ejected as aerosols into the air. And we don't know if that could be involved, but that has been shown for some other strains. Another property that Polaribacter has that I think is quite interesting is gas vacuoles. So that may make them more buoyant. But I have seen this also in the Arctic, these Polaribacter preferentially aerosolizing into the air. So it's very interesting, I think.
Very cool. Yeah, very cool. Thank you. Wonderful talk.
You're welcome. Thank you.
I'm seeing in chat box one question from Stephen Archer, right? Stephen? Stephen Archer, you have one question, right?
Absolutely. Tina, awesome talk as always. I was just more curious about the functional ability of your sampler to collect liquid samples in the Antarctic. Was it always above freezing temperature, or was there a trick that you didn't allude to?
So, I mean, this was all above freezing temperature, but we have also I have used this sampler down to -20 degrees because we use a homemade RNA later solution, which is a 45% ammonium sulfate. And that has a freezing temperature below -20. And I mean, we have tried, we were just in a campaign in this winter in Nuuk, trying to collect at sub-zero conditions, and we got one of these Coriolis samplers. And I mean, this was very difficult to get to work at sub-zero temperatures, while this Kercher impinger, I mean, it's very easy actually. You don't get the same issues.
Oh, I really want to see one of these in operation one day, because yeah, Coriolis seems to, it doesn't matter what the fluid technically can maintain. The RNA later we used was fine at -20. In normal conditions, you get it down to Antarctica, and we're having major issues in the very early negatives. So that's interesting. It must have something to do with how the EMS is actually transferring through. But yeah, amazing.
I mean, the Coriolis also has all these small tubings, and I think we had a lot of problems with freezing with those. And so in the end, we switched back to this Kercher system, which is, I mean, it's a commercial vacuum cleaner, but we did characterize it very well with chamber experiments. And so I'm pretty confident about the cutoff and the performance.
Although the unit I'm really interested in right now is the SAS 3100. If you've seen that, that's really cool. It's filtration based, but it doesn't, no liquid medium, but it seems to get major diversity out of it. But we could talk about that forever, couldn't we?
Thanks, Stephen. Thanks, Tina. I'm seeing another question in question answer box. This is from Aritri Sanel. Thank you for the interesting talk. Did you look into the bacterial composition for the coarse aerosol?
Yeah, so sorry, there was not so much time to go really that much into details, but of course, these data from bacterial aerosols that I show, they are coarse aerosols, because with this impinger, what we collect is above 500 nanometers. So the cutoff is 500 nanometers approximately. So it efficiently collects coarse aerosols, so bacterial cells and bigger. And we did this community composition and quantification of these, as I showed.
Oh, thank you, Tina. Then from my side, one question. You said that this bacterial concentration is low in Arctic compared to the Antarctic, right?
No, it's low in Antarctic.
Sorry, low. Yeah, Antarctic. Yeah. So what may be the reason as?
I mean, I think in terms of terrestrial environments, these are quite low productive systems compared to the Arctic. I mean, there is. I mean, we have heard other talks on this topic, but what we see is that the diversity in the soil samples in Antarctica is not that high. So I think it's also quite interesting that the marine contributions are that high, because that's not what you normally see in bioaerosol populations. Marine aerosolization is considered to be a less dominant source compared to terrestrial sources. But here it's really predominant. So I think it's because of the terrestrial environments are.
Okay, thank you. And another question is that you showed the correlations, temperature and precipitation with bacterial populations, and this is yes, absolutely negative correlations you showed. So is this for any particular community or species, or how it is that?
So these correlations we did with the real-time data, this instrument that I showed. So we get data for every 10 minutes. And then we also have the meteorological data. So it's a high time resolution, but we don't know what these bioaerosols are. We just know that they contain those fluorescent compounds.
Okay, thank you. So any more comments, suggestions from panelists? David, Professor Craig? Okay. So then before closing, I would like to request all to switch on their video so that we'll have a snapshot. Yeah, I request all. And then I would like to ask Dr. Sabu to take a snapshot.
Yeah, this done. Yeah.
Is done?
Yes.
Okay. Thank you.
So
thank you once again to all panelists, speakers, and the IT team who coordinated all for this 2 days since yesterday for this our session, ecological implication and adaptation of microorganisms in polar environments. So I would like to request if anybody have any more suggestions, comments, they can send to us, and we'll try to give this IT team who can go for this YouTube and other media. YouTube, I think, yes, Cary YouTube channel and all. So thank you once again. Thanks. So we can close this session, right?
Thank you.
Yes,
Yes.
we already crossed the time. Yeah.
Thank you.
Bye.
We'll meet
you.
again.
Thanks,
See you.
everyone.
Yeah.
Great to
Thank
see
you.
you.
Yes, see you.
See you.
Bye.
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 Bye.